It's my pleasure to introduce Kara. She came to our lab after graduating at Cal State Northridge, 2014.
Okay.
I asked her, I said, did you take a year off between or something like that? And she's like, nope, I started right after. So yeah, she's been around for a while. And she's been very busy. She's been around long enough to where it's going to be hard for me to imagine our lab without her in it. But it's even better because it won't be, right? I'm going to be able to keep her around for this a little bit. Like if you have office hours and need a place, you can always use my lab space. Anyway, Kara, she started up to revive a project that I had put down for a while whenever I started working on microbial source tracking. And then there was some interest in getting this thing started back up to take a look to see if we could develop a system to express enzymes to take care of the problems that really all of us have with gluten, but many of us have more severe than others, right? So anyway, she graduated, she came up here, started up in the program. She actually walked in 2017. Yeah, but it's just taken her a while to finish up those last little bits there. It's not like she hasn't been doing anything though. Since she's been a master's at Cal Poly, she's actually been teaching primarily these three classes, Bio 111, Micro 221, Micro 224. She's actually taught 2 to 3 lab sections per quarter since she's been here. So a lot of teaching, has had a lot of teaching experience, and actually as a result of that, was awarded the Outstanding Biology Teaching Associate back in 2017 when she graduated. So she's very highly respected from her students and faculty. We know that we can just assign whatever class they're in, she'll be able to take it over and do a great job. So she's very gifted in the lab. She's been helping to train a lot of my students, has done a lot more work than she's going to be presenting today. She's just going to be presenting part of the work that she's actually done in the lab. So she has some abilities. Definitely in the classroom, she's amazing, but it turns out she's also has a superhero. I didn't even recognize until I stalked her a little bit on Facebook. And at first I was like, that's not Kara, no. Oh, it is. One of the other areas. And so she's actively involved in a variety of things. There are tons of pictures of her playing around with our friends in the lab, as well as, I guess she goes to Disneyland a few times, and other places. So she thoroughly enjoys life outside the lab as well. But today we're just going to be talking about what she does in the lab, not her superhero activities. And she's going to be talking a little bit about getting this project up and running. We had a few students before her that had started work on this and were not able to get these proteins to be expressed properly and look at their activity. And so that's where she's started off here, is getting after these enzymes, trying to express them and then evaluate their activity and then look to see if we can choose one of them to be able to develop this treatment. But I don't want to talk about that too much because I'm going to get into her lecture. So it's with honor and a bit of sadness to introduce Kara for her master's thesis defense. And please take it on, Kara.
Thanks for coming today. I'm excited to share with you what I've been working on for the past 5 years. My project centers on developing a protein expression system that can be used in Lactobacillus reuteri to alleviate the symptoms of celiac disease. For today's talk, we're going to go into a background of celiac disease. What is it, how it develops, and then once we get through that, we're going to go into the first stage of the project where we screened through 2 different types of enzymes. to see how they are active at a pH that is relevant to the duodenum of the small intestine. And then in the second phase of the project, once we identified an enzyme that was optimal at that condition, we wanted to incorporate it into an expression cassette for Lactobacillus reuteri. And then finally, we'll end with directions for the future of the study. Celiac disease is an autoimmune disorder that's triggered by gluten. Gluten is a protein found in grains such as wheat, rye, and barley. It can be separated into two types of proteins, one being glutenins and the other being gliadins. Gliadins are going to be the focus of today's topic because they are the proteins that stimulate the autoimmune and immune responses in celiac disease. Celiac disease also affects 1 in 100 individuals in the United States. It primarily affects the duodenum of the small intestine. And if we were to look at a closer look at the duodenum, enlarging one of the villus, celiac disease is primarily going to affect the intestinal epithelial cells as well as the lamina propria. The symptoms of celiac disease include flatulence and diarrhea, abdominal pain, as well as dermatitis herpetiformis. This is a skin rash. More severe symptoms include crypt hyperplasia as well as villus atrophy. To give you an idea of what those last two symptoms look like, this is a cross-section of the duodenum where we have the long finger-like projections, which are the villi, and then the crypts, which are the grooves in between. This is from an individual that does not have celiac disease. Compare that to an individual with celiac disease, you have blunting of the villi, which is villus atrophy, and then elongation of those grooves, which is crypt hyperplasia. Development of villus atrophy results in malabsorption of nutrients for an individual. So now that we have a kind of introduction to what celiac disease is, I'm going to introduce 5 factors that contribute to the development of the autoimmune disorder or response of celiac disease. The first being gliadin. This is one of the peptides of gluten. It's rich in prolines. These proline residues cause the peptide to be relatively resistant to gastric, pancreatic, and brush border enzymes. So within this peptide, the immunogenic epitopes of gliadin are preserved. It's also rich in glutamine residues, which act as targets for tissue transglutaminase. Tissue transglutaminase is going to specifically act upon these 3 consensus sequence within gliadin. And at those consensus sequence, it can make two modifications. The first one being a cross-link, where it will create a peptide bond between a glutamine residue as well as a lysine residue. In celiac disease, tissue transglutaminase can cross-link itself to gliadin. The development of this complex is going to be super important for the autoimmune response in celiac disease. The second modification it can make is deamidating a glutamine residue into a glutamate residue. The transition from a polar uncharged amino acid to a negatively charged amino acid in this peptide changes its binding affinity to these MHC class II molecules, specifically the HLA-DQ2 or DQ8 variants of that molecule. These MHC class II molecules are responsible for presenting these immunogenic peptides or epitopes to T cells. This specific variant of the MHC class II molecules have a binding preference for negatively charged epitopes. So these molecules will preferentially present deamidated gliadin peptides to our T cells. And then the last 2 contributors are going to be our autoreactive tissue transglutaminase specific B cells as well as a leaky gut. And altogether, these are going to initiate the autoimmune response. And so here we're looking at a section of the duodenum. At the top, we have the lumen with our gliadin peptides, and then our intestinal epithelial cells with the X's representing our tight junctions, and then the lamina propria right below it with our autoreactive tissue transglutaminase specific B cells, as well as a gliadin specific T cell. Under normal conditions, these tight junctions prevent the passage of macromolecules from the lumen into the lamina propria. But in the presence of gliadin, these tight junctions increase impermeability so that these gliadin molecules will be able to enter into the lamina propria. Once inside the lamina propria, tissue transglutaminase will be able to cross-link itself to gliadin and also deaminate that peptide. This complex will then be recognized by our tissue transglutaminase specific B cells, and it'll be internalized. And inside that B cell, it will be degraded and processed, and then the B cell will be able to present both deaminated gliadin as well as tissue transglutaminase by those MHC class II molecules that we talked about previously. When they are presenting these deaminated gliadin peptides, our gliadin-specific T cells will be able to recognize that and stimulate the B cell to produce anti-tissue transglutaminase antibodies. These antibodies will then be able to recognize tissue transglutaminase, and altogether they cause the autoimmune response to take place in celiac disease. There's currently no cure for celiac disease. However, the only treatment for celiac disease is to maintain a gluten-free diet. There have, however, been biotech companies that have started to produce oral enzyme therapies that will allow an individual to break down, or not the individual, but the enzyme can break down gliadin within the stomach. These two treatments, however, don't address the issue of accidental gluten exposure. So that's where our proposal comes into play. We're proposing the idea of having a probiotic colonize the duodenum, and at the site of the duodenum, these probiotics will be able to synthesize and secrete an enzyme that will be able to break down any gluten that is encountered in the duodenum. Which now leads us into the first stage of our project. In this first phase, we screened through 2 different types of enzymes and assessed their activity at a pH that was reflective of the duodenum. We accomplished that by doing codon optimization, protein expression, and purification in E. coli, as well as assessing enzyme activity through the use of chromogenic substrates. In this study, we used prolyl aminopeptidases, which are enzymes that would cleave adjacent to a terminal proline residue, as well as prolyl endopeptidases, which cleave adjacent to internal proline residues. The 2 enzymes we have, we had 2 PAPs that were derived from Lactobacillus acidophilus and Lactobacillus reuteri, and then we had 2 PEPs that derived from Aspergillus niger and Myxococcus xanthus. Because we have these enzymes originating from 4 different organisms, we may run into the issue of codon bias. Because the genetic code is redundant, meaning that you have multiple codons representing a single amino acid, one organism may utilize one codon at a higher frequency than another organism does for that same amino acid. And that's what codon bias is. And so, in order to compensate from codon bias, we could use codon usage table frequencies to optimize our sequences for expression in Lactobacillus reuteri and E. coli to eliminate the problem of codon bias. Here we're looking at a codon usage table for 6 arginine codons in Aspergillus niger, E. coli, and L. reuteri. These values represent the frequency of each codon among all the arginine codons that are used within that organism. And as you can see, at this bottom codon, AGG, it's used relatively infrequently between E. coli and Lactobacillus reuteri. compared to this codon above, CGU, where you see a 10-fold increase in the frequency for that codon. So by using codons with a higher frequency, we can rewrite the sequence to eliminate codon bias. In this image, we have a section of the original Aspergillus niger PEP, and we are looking at the codon usage frequency within Lactobacillus reuteri. So here we have the original Aspergillus niger codons, and these bars are going to represent the frequency of those codons in Lactobacillus reuteri. But specifically, these bars represent relative adaptiveness values. Those in relative adaptiveness, the codon with the highest frequency is set to a value of 100, and then the remaining codons for that same amino acid are going to be scaled into relation to that. And so you could see in this table that some of the codons that are from Aspergillus niger are used relatively infrequently in Lactobacillus reuteri, as signified by these red bars and these gray bars. So what we can do is rewrite the sequence, incorporate codons that are used at a higher frequency to eliminate codon bias. Codon bias can become an issue because it can slow down or halt the rate of translation, so we can eliminate that issue by optimizing the sequence. We optimize the sequences for Aspergillus niger PEP and Myxococcus xanthus PEP. We didn't need to optimize the sequences for the LA-PAP and LR-PAP. Once we had our final sequences, we incorporated them into a PEP30 expression vector for E. coli. This expression vector had a histidine tag that allowed us to purify our proteins of interest. Once we constructed those vectors, we transformed them into E. coli BL21, and then grew up those cultures in LB broth to an OD 600 of 0.5. Once they reached that OD level, we separated them into two samples. One was induced with ITPG, and the other was not induced. This sample that was induced with IPTG would cause our cells to produce our enzyme of interest. This bottom culture was not induced, so they would not produce our protein of interest. So at the end of this overnight incubation, we have a culture with our protein of interest and then a control culture without our protein of interest. And so for the rest of these methods, I'm going to just continue with this induced culture just to make things more simple. Once we had our culture, they were centrifuged, the supernatant was discarded, and the cells were resuspended. These resuspended cells were then sonicated to lyse open the cells. Once we had those lysed cells, we separated them into two fractions, one being the lysate, which should contain our soluble proteins, and then the pellet, which should contain insoluble proteins as well as cell debris. We ran these lysates out on a nickel column to purify our protein of interest from all the other cell proteins. Once we had that purified sample, we ran them out on SDS page gels. In this first image, I'm going to orient you to what these lanes are, and it's going to repeat for the following gel images. So at the top, we have whether or not that sample was induced with IPTG. The minus sign was not induced, so we should not expect our protein of interest. The plus sign was induced, so we are expecting the protein of interest to be there. And then these first two lanes are going to represent total protein found in the sample, and the last two are going to represent our purified samples. So with the LA-PAP, you can see that it is being detected and purified at the expected band size of 38 kilodaltons. With our MX-PAP, we're seeing a similar thing where it's purified at the expected band size of 79 kilodaltons. It's probably in this total protein sample, but because it's such a high molecular weight, it's going to be hard to differentiate between the other proteins within the sample. And then the LR-PAP tells us a similar thing. It's being detected and purified at the expected band size of 37 kilodaltons. Now, we're going to get into the problem of these enzymes. Our ANPEP did not express, or was not expressed as a soluble protein, as you can see by nothing being here. So because we didn't see ANPEP expressed as a soluble protein, we looked back at the pellets, which should contain our insoluble proteins, to see if it was still being produced. And as you can see, with this induced sample here and this induced sample here, we are seeing bands that are unique to those induced cultures. And specifically, I want to point out these 3 bands. There's one at around 55, 40, and around 15 kilodaltons that are detected in the total protein samples, and then purified as well, those same bands. These bands suggest that ANPEP is being produced as an insoluble protein, and it's also being cleaved into fragments. In addition to those 3 bands, we also very faintly see one band around 60 kilodaltons. We're expecting our Aspergillus niger PEP to be 63 kilodaltons. So that band suggests that we are producing the full length Aspergillus niger PEP, but also as an insoluble form. From these, the data that was presented here, we did not continue on with our Aspergillus niger PEP because we couldn't purify it as a soluble protein. And so for our enzymatic activity assays, we continued on with our LA-PAP, LR-PAP, and MX-PEP. In our reactions, all of them contained 100 millimolar phosphate buffer at pH 7, which should represent the pH of the duodenum. And then they also contain chromogenic substrate. The MX-PEP contains the chromogenic substrate succ-alipro, which would simulate the internal proline residue. Suc is a protective group during the synthesis of that peptide, so that proline and alanine kind of link together in that order. And then you have our PNA, or our p-nitroanilide, which is our chromophore. When it's released from that substrate, it should turn yellow. H-Pro is emulating our terminal proline residue. This H is just a hydrogen, and then you have your PNA, which should also be cleaved and then turn yellow. And then finally, we have our enzymes in these reaction volumes. The absorbances of these reactions were measured at 410 nanometers at 37 degrees Celsius. And the absorbances were measured every 30 seconds for 10 minutes for the MX-PEP and succ-alipro reactions. For the LA-PAP and LR-PAP, the absorbances were read every 30 seconds for 60 minutes. After collecting this data, we were able to calculate Km, Kcat, and Kcat over Km for all our enzymes. Because we were using LA-PAP and LR-PAP against the same chromogenic substrate, we could make a general comparison between the two. So for the first value, we calculated Km. Km is going to be an indicator for the enzyme's affinity for the substrate. And so a lower Km value represents a high affinity for the substrate. Here we can see that LA-PAP has a slightly higher affinity for H-Pro than LR-PAP does. For Kcat, that is the rate of the reaction, so how fast it's cleaving that H-Pro PNA substrate. And with the higher Kcat value, we can see that LR-PAP has a higher reaction rate than the LA-PAP. And then finally, Kcat over Km is the enzyme specificity for the substrate, or its preference for one substrate. And then again here, we can see that LR-PAP has a higher enzyme specificity than does the LA-PAP. With these two enzymes, there weren't any other studies that made a comparison or like assessments between these enzymes against that same chromogenic substrate. So we can't really say whether or not they were like highly efficient or highly ineffective against cleaving that substrate. We just know that it did cleave it, and then we can make the comparison between these two enzymes where the LR PAP might be slightly more effective than the LA PAP against that substrate. With our MX PEP, we did have 2 studies to make the comparison to. We have a group from 2012 or 2002 and 2017, and we all calculated the same values. So for KM, our value was higher, so that suggests our MX PEP had a lower affinity for our suc-allopro substrate than these previous 2 studies. With KCAT, our reaction rate falls in between the 2, but slightly on the lower side. And then for KCAT over KM, we see a similar trend. It still falls in between the 2, but again on the lower side. So from this comparison, we can probably conclude that our MX PEP is not the most efficient MX PEP out there, but it is comparable since it has comparable results to this Kogadag group. From this study, or this portion of our study, we saw that AN PEP was produced as an insoluble protein. That may be linked to codon optimization. There has been a study that has found that if you substitute the infrequent codons for the more frequent codons, you can change the rate of translation, so going from a slower rate to a faster rate. That change in rate may affect the protein's ability to fold correctly. If you're going too fast and it needs to slow down, it's not going to be able to fold correctly, and that may decrease solubility of the protein. So that may be a reason why we're seeing AN PEP expressed as an insoluble protein. With the LA PAP and LR PAP, we did see cleavage activity against our terminal proline residue, or substrate. However, it was not effective, or we didn't see activity against the internal proline residue substrate. And so for that reason, this enzyme, or these enzymes, may not be our primary candidate to move on to the next stage of the study. Gliadin has many proline residues within the peptide and not just on the ends. And so these enzymes would not be effective at cleaving the immunogenic epitopes fully of gliadin. What we could do is use these enzymes in tandem with another enzyme that could break down the bigger peptide, and then these enzymes may be able to break down any terminal proline residues that may show up. So they can be used in tandem, but not as the primary enzyme candidate. After screening through these 4 enzymes, MX-PEP seems to have the best characteristics to go on into the next stage of the study. It was capable of cleaving adjacent to internal proline residues. It wasn't the most effective, but it had comparable activity. And it also has been shown to have optimal activity at a pH of 7, which is similar to that of the duodenum. And for these reasons, we used MX-PEP and tried to incorporate it into an expression cassette for Lactobacillus reuteri. In this section of our study, we're going to look at, briefly, why we chose Lactobacillus reuteri as a delivery vehicle. We'll also go into the construction of the expression vectors that were used for expression in Lactobacillus reuteri, and then look at cytosolic protein expression. Lactobacillus reuteri is a probiotic, which generally are defined as live microbial organisms that are capable of influencing the gut microbiota and also conferring health benefits to a host if they are applied in appropriate amounts. These probiotics are also capable of surviving gastric acid, bile salts, and pancreatic juices. This is an important characteristic if we want our delivery vehicle to end up in the duodenum. In order for the delivery vehicle to get there, it has to go through the stomach, so survive those gastric acids. And then in the duodenum, there's a dumpage of bile salts and pancreatic juices. And so if it can tolerate those conditions, it should be able potentially to colonize that area. Lactobacillus reuteri specifically is endogenous to the duodenum, so we know it's going to colonize that area. So it makes it a good candidate already. Moreover, it has been shown to outcompete foreign probiotics for colonization. These organisms are capable of synthesizing an antimicrobial compound called ruterin, which has been shown to have inhibitory effects on enteric pathogens as well as other commensal bacteria. So it has the potential to become a prominent member of the duodenum. And then for those reasons, we chose Lactobacillus reuteri as the delivery vehicle to deliver MXPEP into the duodenum. For expression of MXPEP, we use these PGK-MCS expression or shuttle vectors. These vectors all contain an erythromycin promoter for constitutive expression of MXPEP, as well as a histidine tag, so we could purify the enzymes, and then a ripple terminator to terminate transcription. In addition to that, we also made a GFP version as a control group for this vector. These two vectors would generate cytosolic versions of these two enzymes, or proteins. In addition to those vectors, we constructed a vector with an amylase signal sequence and put that right in front of our MXPEP gene. We wanted to use the signal sequence to generate a secreted version of the protein. This amylase signal sequence was chosen because a previous study had shown that it was successfully used to secrete GFP into the extracellular space, and they used it in Lactobacillus reuteri. And then we also created a GFP version of this vector. After constructing all these vectors and transforming into E. coli, we ran into a few bumps in the road when we were looking at our transformants, particularly with our cytosolic GFP vector. When we're doing these transformations, if they're getting potentially the same piece of DNA, we should expect to see that they all have the same phenotype. That's not what we saw with our transformants. We saw 3 phenotypic variations from our cells. These light cream colored ones are most likely cells that took up a self-ligated version of the vector, so it did not contain GFP. But the big kicker was these 2 variations in the GFP expression. So we see one that has an intense green color, and then one that has a dull green color. When we looked to see what vectors these colonies had, we found that the dull green colonies had GFP oriented in the same direction relative to our erythromycin promoter. And that's where we were expecting to see that GFP was being expressed. But what we did not expect to see was the more intense green colonies to have GFP oriented in the opposite direction of our erythromycin promoter. So, what's happening? When we looked further into the construction of these PGK-MCS vectors, we found out that the MCS contained the lac promoter. So what we were seeing was there was expression from the erythromycin promoter when GFP was oriented in the same direction, but this lac promoter was somehow overpowering the erythromycin promoter and driving stronger expression of our GFP. And so when we have these expression vectors, we don't want to have any competition between 2 different vectors. We want ours to be the best. And so in order to be the best, we have to eliminate the competition. So we took out the lac promoter to construct these versions of our vectors. We did it for the cytosolic GFP and MXPEP, as well as the secreted MXPEP. And so at this stage, when you've constructed your vectors for a second time, you think that you can't get any worse than this. But that's where you're wrong. We had a ton of issues with this secreted MXPEP. When we transform these vectors into E. coli, we normally do a diagnostic digest on them to confirm that the vector is in the cells. With this vector, we did a diagnostic digest with XHO1, there's a site here and a site here, and then NCO1. What we are expecting to see are these 3 fragments showing up from diagnostic digest of this vector. But when we screen through 8 different colonies, and get ready for this, we only saw 2 of those colonies showing up with the expected band patterns. The remaining 6 either had a completely different pattern or slightly similar, but still not what we were expecting it to be. So it appeared as though there might be some selective pressure that led to the accumulation of mutations within this culture. And because of the instability within these vectors, we wanted to look back at all 6 of our vectors to see if they would remain stable over a longer period of time. So what we did with these 6 cultures is we isolated a colony and then subcultured that colony for a span of 5 days. At each day, we isolated those vectors and then did diagnostic digests, again, XHO1 and NCO1. With our cytosolic GFP vector, here's a graphic map of those restriction sites and what fragments should show up from those digestions. We see that the patterns, it's hard to see, but these patterns are the same between all 5 days. These little bands at the top are probably due to incomplete digestion of the vector. Yeah, that's better. So you can see that very faintly, you get both of those bands at the bottom. When we take out the lac promoter, again, graphic map showing the expected band sizes, we see that these 2 bands still remain as expected. We still have that 4300 band, but then by the 3rd day of subculturing, we see the development of another band in our digest. With our cytosolic MXPEP vectors, graphic map again, and band fragments, we are seeing that all these digestion patterns are consistent with each other over the 5-day period, and the expected band sizes are showing up. The only thing is we're getting this extra band at around 3,000 or 4,000-ish. That may also be due to incomplete cutting. But what the takeaway is that the band patterns are all consistent. And then when we take out the lac promoter from that vector, by day 4, we can see that these 2 bands develop and that 1992 band that should be around here disappeared. With our last vector, this is where things get really wild. Over the 5-day period, this vector, the secreted MXPEP vector with the lac promoter still, all the band patterns are consistent. When we take out the lac promoter, we see that the 1st day we are getting the expected band patterns, but then by the 5th day, the band pattern completely changes. These data suggest that by removing the lac promoter, we are causing the vector to become unstable or destabilizes the vector, especially with the vectors with the signal sequence and MXPEP. So because of this instability with these vectors, we didn't want to move on with expression of those vectors in Lactobacillus reuteri. So instead, we went back to our original vectors that still had the lac promoter and assessed protein expression, but in E. coli. So what we have here is a Western blot from our cytosolic GFP vector. And we have the lanes that are labeled with P, those are our pellets or our insoluble protein fractions, and then lysates, which should be the soluble protein fractions. And then the first two lanes are total bacteria, and then the last two are going to be our purified. So this was probed with anti-GFP. We can see that GFP is being detected at the expected band size of 30 kilodaltons. In our total protein lysate, we are seeing the expected band size right here, but then we also see a band at lower molecular weight. It looks like GFP is getting cleaved, and potentially the his tag might be getting cleaved from that protein. And then with our cytosolic MX PEP, we are seeing it detected at 79 kilodaltons. So these data show that our expression, or our vectors, are successfully working in E. coli. The big issue is to get it to express in Lactobacillus reuteri. And we transform these into Lactobacillus reuteri, these two vectors. And the biggest disappointment of it all is that they did not express GFP nor MX PEP. So when you don't see that, you have to keep searching for answers. And so we did that by sequencing our vector. We did this for our two cytosolic vectors, the GFP and the MX PEP version. The sequence data revealed that this erythromycin promoter was the same sequence as a previous study that used it and saw expression from Lactobacillus reuteri, and they were expressing GFP. So we know that this promoter is likely not the explanation as to why we were not seeing protein expression in our probiotic. And then also GFP was the same sequence as we were expecting it to be. So those two sequences were not to blame for the lack of expression. With our Myxococcus PEP sequence data, we didn't get any sequence data from that, and that's most likely because I used the wrong primer. So we didn't get any data telling us about whether the MX PEP sequence was good or not. But regardless of that, we have protein expression from this vector. We know that this sequence is not mutated or anything. And so the sequence itself is not to blame for the lack of expression. So we look further into why we're not seeing expression in Lactobacillus reuteri. And what we found is this space in between the promoter and the start codon of GFP heavily influences translation efficiency. So what we're looking at here is part of the cytosolic GFP vector. This is the 3' end of our erythromycin promoter right here. And then ASC1 site that was used to clone in GFP, and then our start codon of GFP. Normally, what happens in translation is you'll have the 30S ribosomal subunit come in and find that ribosomal binding site, and then once it finds it, it should be able to initiate translation. But the tRNA is going to enter the 30S ribosomal subunit at the P site. And as you can see, the start codon is not there. So what we discovered is that the space between the ribosomal binding site and the start codon plays a big part in whether translation will initiate. And we found out that we know that our spacer, what we'll call that space in between those two sites, is 11 nucleotides. And the optimal length of the spacer for optimal expression in Lactobacillus reuteri is between 7 and 8 nucleotides. So ours is too long. What Jared did, who was a previous grad student in our lab, is he took out this ASC1 site and replaced it with EcoR1 and shortened it. So it goes from 11 nucleotides, oops. tRNA doesn't know what to do because it doesn't see the start codon. So Jared replaced that ASC1 site with this EcoR1 site and shortened it from 11 nucleotides to 9 nucleotides. So it's not optimal, but now translation should initiate. And what Jared found out is when he did that switch, he did see expression of MXPEP in Lactobacillus reuteri. So all of that was to blame for that small little space in between the ribosomal binding site and our start codon. So from our study, we can make 2 overall conclusions in this part of our project. We saw instability with our PGK MCS expression vectors when we took out the lac promoter. One of the main reasons I think is that this erythromycin promoter is a constitutive promoter, so expression is always on. What we can do instead is use an inducible promoter to switch expression on and off, and hopefully that will stabilize these vectors in E. coli. The second is that we did not see any recombinant protein expression in E. coli or Lactobacillus reuteri, and we know now that it's because the spacer in between the ribosomal binding site and the start codon was too big. So for the future of this study, we have a CLIP promoter, which is an inducible promoter that we can use in E. coli to turn expression on or off and hopefully stabilize the vectors that then can be transformed into Lactobacillus reuteri. And then one other major thing that we want to do, the ultimate goal of this is to have secreted version of our MXPEP. One thing we might want to try is to screen through more signal peptides. Even though our amylase signal peptide has been shown to effectively secrete GFP into the extracellular space, other studies have shown that a signal peptide that works well for one protein may not have the same effect on a different protein. So that amylase signal peptide that works well for GFP may be ineffective with MXPEP. So we need to screen through more signal peptides, test them solely with MXPEP, or just like a portion of MXPEP to see how secretion efficiency is with those peptides, and then use that one as our final peptide for this project. So I want to thank my committee for rushing to get to this point. I know we had a very small amount of time to schedule this. I'm very thankful that we were able to do this before Dr. Hillers left. And I also like to thank my advisor that I've had for the past 5 years. I can do that too. Just thank you guys for working with me.
From last year.
I'd also like to thank faculty that have worked with me throughout my time at Cal Poly. And then also staff, the Brewster brothers, Dave, Alice, John, for always helping me whenever I need something or can't find something. And then biology office, Ellen, Melanie, Kristen. And then Black Lab, Joseph's here. And then the past grad students that have been here that have carried me on through my time at Cal Poly. And then all the babies that were born when I was a grad student. And yeah. Any questions you guys have? Yes, Dr. Keeling.
So you mentioned at the end this idea of this inducible promoter.
Yes.
And you said it's inducible in E. coli. Is it not also inducible in Lactobacillus?
So that CLIP promoter that we have, it's inducible by heat or like any type of push stressor. And so as long as the repressor is present in the organism, we can induce expression. I know for reuteri, it already has the repressor, but for E. coli, it didn't. But we have like a vector with the repressor, so we can now induce expression in E. coli as well,
Or at least shut it down.
or shut it down.
But so the idea is ultimately you want it in the lactobacillus rear
Yeah.
eye, right?
Mhm.
And in the rear eye, do you want it to be inducible or do you want it to be constitutive, right? Because if someone's
Yeah.
gonna take this, it's
Yeah.
gonna be inside them.
Mhm.
Seems like you might want it constitutive. But if
Yes.
it's gonna be unstable when you make it be on all the time,
Right.
then wouldn't that be a problem for
Yeah.
having it actually be useful?
Yeah, I guess it depends on, when we get to that point, like, if we're seeing constitutive expression in Reuteri and it's not having any adverse effects on it, then we could maybe keep it around, because that would probably be the ultimate goal of it, is to have it always there so that if gluten is encountered at any time, it could be there to break it down. But I think for the time being, like in the lab setting, being able to shut it on and off is probably a better idea, because serine the Myxococcus pep is a serine protease, and those are really active and like very kind of almost, I don't know what the word is, like not explosive, but they're very like, they like to cut things. And so,
But then wouldn't you worry about that if it was a probiotic inside a human? I mean,
yeah.
if your serine proteases
Right.
are highly reactive and are gonna cut random things.
Mhm. I guess that also depends on like the specificity of the enzyme. We only tested it on the chromogenic substrate. We don't know how effective it can potentially be against like the actual peptide itself or other proteins. So that's probably something we could also look into, is to how it affects other proteins that may be around.
Just follow one thing also that she's running into. The biggest problem is we can't get it into Reuteri. If we can't get it through E. coli, right, then we can never test it. So we have to have something at least to get through E. coli
Sure.
and then be able to test it out. But it's a good question.
Absolutely.
Yeah, Jen.
So you had your initial 3 phenotypes of your transformants, the white, okay,
Yeah,
and then your light green and your bright green.
you're right.
And the light green had the GFP in the right orientation for the promoter, which is what you wanted. So why didn't you just follow up with those? Were they just not expressing enough protein to follow up? Or was because you wouldn't expect the GFP or whatever protein is in there is to switch and then be going from the lactose promoter, right? Like,
I think it's because we wanted to take the lac promoter out so that expression from the promoter wasn't like dulled by the lac promoter. And you don't want to have like this conflict where you have 2 promoters going in opposite directions and then like creating whatever product. So if you remove one, you should have like expression only coming from a single promoter and not like 2 different
okay,
ones.
so the lactose promoter, even though it's making nothing,
Well, it was
it's not making your GFP if you know, the
I see what
GFP in
you're
that
saying.
direction,
Yeah.
right? It's coming from the other direction, so
Right.
it's making an anti-GFP, if you
Yeah,
will, whatever,
yeah.
a junk protein.
Yeah.
But it could compete,
Yeah,
is what you're saying?
I think one of the reasons why is we saw like a difference in intensity, where the lac promoter was showing more GFP expression compared to the promoter. And so we thought it was overpowering it, and so whatever, even though like GFP could be in the same orientation as the promoter, whatever is coming from the lac promoter might have some negative effects on like expression from that promoter.
Aren't there plasmids that have lac repressor?
Yeah. When I got the PGK MCS vector, I didn't know that the lac promoter was there. So I think the easiest thing for us after we figured it out was just to
Get rid of it.
get rid of it.
But then that created an
Yeah.
unstable plasmid.
Yeah, there's like, you do one thing and you
That's
think it's the
molecular
right
biology.
thing and then it all goes, yeah.
Yeah.
What do you think is just to follow up on that, what do you think is causing the instability? Like why is it removing this, or what can you speculate as to being
What?
result
Well,
of that?
if it's making like the MX pep transcripts and those just happen to be toxic
Right.
to the cell, it might want to stop that from happening by mutating or like selecting for mutations that stop those transcripts from accumulating.
Okay, I'm gonna ask a follow-up question on that. As you did those time courses,
Yes.
did you just cut with a single enzyme? Like,
Uh,
were there pieces that were popping out because of maybe recombination within the plasmid, because sequences were removed and you got now some homology that, I don't know, I'm just, but yeah.
We did a double digest with 2
right,
enzymes.
you were seeing that new band coming up.
Oh, right.
So is your plasmid just like?
Yeah, it could be doing some weird things that we can't really detect for, because it's just a very simple diagnostic digest, and so
Right.
if that site is there, it'll cut, but
Right.
if it's not there, other than that, we can't really know what it's doing.
Right.
Okay, I have a question, back to celiac disease
Yes.
and these prolyl endopeptidases.
Mhm.
So, do we make PEP enzymes, or do only bacteria make PEP enzymes? And
Yeah.
in normal people versus celiac? Sorry, I'm sorry.
Oh.
I'm sorry, I misspoke.
No, you didn't. No, I don't think you did. I think
Not
you're right.
celiac disease, and people who do have celiac
Yeah.
disease have different levels of PEP expression.
normal people.
Different microbiota. What do we know about that? I apologize.
It's okay.
I know in humans, they have found PEPs in the brain that affect, or like they have some sort of contribution to Alzheimer's, but I think they're absent from the gastrointestinal tract.
Okay. So we
Yeah.
are dependent on our microbiota for PEPs in our
In-
GI tract.
Yeah, in that sense,
Okay.
I would say, yeah.
So have people looked to see if there are differences in PEP levels in people with celiacs versus not in the gut? I mean,
Yeah.
produced by their endogenous microbiota.
I
Like,
have
why
not,
aren't these
yeah.
things being cut up in general? Or are they?
Well, I guess it depends on the organisms in the gut microbiota,
Mhm.
because probiotics like Lactobacillus, Acidophilus, and Reuteri, they make PAPs. They don't make PEPs. So that might affect, that might be why.
So PEPs are not made by normal gut microbes,
Well,
typical gut microbes?
not that I have
Or secreted,
seen,
maybe.
yeah. Oh, yeah, secreted,
Right.
yeah.
Mhm.
They're made, but maybe not
But
secreted.
not secreted.
Just to kind of follow on that, how long have we been eating gluten? How long has wheat
A lot.
and barley and rye been in the human diet? I mean, we're talking about, the evolution of
Yeah.
enzymes, like, these are the primary grains and in terms of agriculture and what our bodies are used to. We did not evolve in a wheat gluten containing environment. Eating, I mean, that was something within the last 10 to 15,000 years, I think. They've suggested that it's gotten into the diet, right? So, so we don't have enzymes that deal with this, yeah. It's the next stage of evolution, really.
Ben?
Has there been anything any research out there to kind of evaluate the immunogenicity of these gluten peptides, like even in the presence of PAPs, like do PAPs, even though they only cleave, at termini, do they still have the ability to decrease immunogenicity and kind of alleviate some of the phenotypic?
Great question. The only like studies that I've seen have used PEPs. Yeah.
Great. So we'll have a meeting here probably within the next 5 or 10 minutes, so please go over and enjoy some tea and treats and stuff like that. Congratulate Kara. And then when you guys are all done, go ahead and head on out and we'll have our last meeting.
How did it go?